Welcome back, guys. So we're at Concept 7.2. It says membrane structure results in selective permeability. As we kind of talked about when we did the chapter overview, the membrane allows certain things in and certain things out, but it is selective. It doesn't just allow anything to enter and exit the cell, and not everything enters and exits the cell at the same speed or at the same rate. So for a cell to be alive, it must exchange certain materials with its surroundings. It has to get food and oxygen, it has to get rid of waste, carbon dioxide, and those types of materials, and there has to be water and ions and other chemicals and molecules entering the cell and leaving the cell. But that doesn't mean that the cell has to have an open door policy that allows anything in and out. So it is what we call selectively permeable, which means that it does regulate the traffic in and out. The textbook also mentions how these biological membranes are a great example of what it calls supramolecular structure, which means there are many molecules ordered together to form a higher function. And so the cell membrane has these what we call emergent properties. Again, you've heard that phrase quite a bit so far in class, where we have levels of organization from the very small all the way up to the very large, and at various levels of organization we have different properties that come about. So a phospholipid all by itself can't do this job, but phospholipids arranged in a certain way in conjunction with certain proteins can do the job of the cell membrane. So this would be a supramolecular structure where you have many molecules that all work together in an organized fashion to do something that one of these molecules alone could never do. Different materials can get through the lipid bilayer in different ways. Hydrophobic, nonpolar molecules such as hydrocarbons, carbon dioxide, oxygen, they have the ability to either they're small enough or they are alike enough, nonpolar, they can kind of dissolve through this lipid bilayer and pass through the membrane fairly quickly on their own. Other molecules that are polar molecules, or larger molecules, such as sugars, and even smaller molecules such as water, if they're polar molecules, they can't easily get across this membrane. They don't have an easy time crossing this lipid bilayer. That center portion of the bilayer, the hydrophobic fatty acid chain tails, they have a hard time getting through all of that. So what we find is that all these various proteins that we talked about, the proteins that are embedded in the cell membrane, here is one of their very important jobs, getting material in and out of the cell. There are some things that can pass through the membrane all by themselves, but for the most part, a lot of the other molecules that we're going to talk about, they use a protein as a pathway to get through this lipid bilayer. Here's a quick little animation of membrane selectivity. So you can right-click on this and hit play, and it kind of shows you what we're talking about. So if you're a molecule that can't easily get through the membrane all by yourself, you're going to need a little bit of help. And so you're going to rely on what's called transport proteins. Transport proteins allow hydrophilic substances to get across this hydrophobic fatty acid tail lipid bilayer. So the transport proteins usually do this in a couple of different ways. Some transport proteins basically dig a tunnel. You can think of it as a tunnel. The protein spans the entire membrane, and through the middle of the membrane is a tunnel or a channel that is hydrophilic, and that gives a pathway for these hydrophilic molecules to pass through. If you've ever driven on vacation up in the mountains, sometimes there are tunnels that travel right through the mountain. Now, there's a different way to do that. You could get out of your car and grab a shovel and start digging your way through the mountain itself, and you might eventually make it out the other side, but it would be long and difficult, and it would take you quite a little while. So the channel or the tunnel allows for you to pass through that mountain quite a bit faster. That's basically what the transport proteins do in this case. They provide a channel or a tunnel for certain molecules to pass through the lipid bilayer very quickly and easily. Certain molecules and certain ions kind of have to have this tunnel to get through in any fast fashion. One of the major things that has to get through is water. And water can pass through the cell membrane on its own, but oftentimes it uses a particular transport protein, and we call these aquaporins. Aqua having to do with water, of course. So the channel proteins that allow water to go through are called aquaporins. And each aquaporin can allow up to 3 billion water molecules per second to pass through. And that's way faster and easier than the water trying to get through the cell membrane all on its own. The other major kind of protein is what's called a carrier protein. And the carrier protein actually does a bit more than just provide a path or a channel. The carrier protein actually binds to the molecule that needs to move across, and it changes its own shape and allows them to cross that way. So we'll see an animation of this in just a moment, but a carrier protein is a type of transport protein that does more than just provides a pathway. It binds to the molecule, changes shape, and that allows the molecule then to get to the other side. And this type of transport protein, called a carrier protein, is very specific to the type of substance that it will allow through. So it's like a helper that will allow you to get across this barrier, but it's very specific. It usually will only attach to or combine with certain molecules. And a good example is in the plasma membrane of your red blood cells, there's a carrier protein for glucose, and it can transport the glucose molecule across the barrier 50,000 times faster than glucose can make it across the barrier on its own. But this protein, this transport protein, is so selective that it will basically only attach to glucose. It won't even attach to fructose. And if you'll remember, fructose is exactly the same as glucose with a slightly different structure. It's a structural isomer, but even that is too much of a change. So a lot of these transport proteins called carrier proteins are very specific. They will attach to a very specific molecule and then change shape, and that allows the molecule to then get across the membrane and into the cell. So transport proteins are very important to get material in and out of the cell, and they do it two different ways. We have channel proteins, which basically just provide a tunnel or a pathway, and then we have carrier proteins, which actually attach to the molecule and change shape, and that allows the molecule to get inside. But what we don't know is what decides the direction of travel. In other words, how do we know, or what determines if a glucose molecule or a water molecule is going in or out of the cell? I mean, it's not like they have brains. It's not like they make this decision on their own. So what is the mechanism that drives molecules out of the cell? Or what is the mechanism that drives molecules to enter the cell? So what we need to talk about is what we call passive and active transport. And we'll get into diffusion and osmosis. So Concept 7.3 will help answer, why do molecules move into the cell, or why do they move out of the cell? And we're going to talk about that in two different ways, passive and active transport. And the first one we're going to go with is passive. And based on that word, you can tell that there's probably not a lot of labor and effort involved here. Concept 7.3 says, passive transport is diffusion of a substance across a membrane with no energy investment. And notice I underline no energy. So passive transport requires no work or energy investment by the cell to get this done. And the word for this is diffusion. And diffusion is the tendency for molecules to spread out evenly into a space. If you were in a big empty classroom and in one corner of the classroom you opened a perfume bottle and you had students that were sitting in the far opposite diagonal corner from that perfume bottle, it may take a while, but slowly those perfume molecules will start to diffuse through the air, and they will spread until they become uniformly distributed throughout the entire room. And eventually, even though you're in the far corner, you will smell the perfume as it diffuses, those molecules diffuse throughout the air. So, although each perfume molecule has no direction, it's random. It could be going up, down, left, or right. Over time, the population of molecules does tend to be directional. It would be out and away from the bottle, spreading through the room. So eventually, it would reach what we call an equilibrium, a dynamic equilibrium. So you have molecules that are evenly spread everywhere, and in the case of a cell, you have as many molecules leaving the cell as you have entering the cell. We want to keep in mind that when I say equilibrium, I don't mean that there is no motion in or out of the cell, that there is no molecules crossing the membrane. There are. Equilibrium just means that the net movement in and out is equal. In other words, however many water molecules coming in, you have about that many going out. So that's equilibrium. There's still motion in and out of the cell, but it's going to be net movement of 0. You're still going to have about the same number going in as you have going out. Here's a quick little animation about diffusion. So if you want to hit right click and play, you can watch this quick little animation about diffusion, how molecules will spread evenly, eventually evenly throughout the area. Let's take a look at a quick example. So here in this example, and you could set this up in the laboratory very easily. Let's say you have a container of water, and it's separated in the middle right here by a synthetic membrane, a man-made membrane that allows selective permeability. And so this is entirely filled with water. On the left side of the membrane, you add food coloring or dye of some kind. So these little balls represent the molecules of dye. So when you start, there's nothing but pure water on the right, and there's dyed water with the various dye molecules, food coloring molecules on the left. Over time, you're going to start to have diffusion. As these molecules spread, they're going to start to enter the right-hand side, and eventually, over time, you would have them reach equilibrium. That doesn't mean that the molecules are no longer moving. It just means that now they're evenly spread, and as many molecules as you have going left, you have going right. So whereas if this were, say, a purple color, you might have it strong purple color and clear water on the right. Well, then as time goes by, you'll end up with a container filled with light purple colored water as these molecules have diffused evenly and eventually reached what we call equilibrium. What if we were to do that with two different colors of dye? Well, what you'll find is that the same process occurs, and it recurs completely separate. They're unaffected by each other. So, for example, let's say, and we'll just use the colors they have. Let's say we have water in a container that's separated by a membrane, and on the left we have a heavy amount of yellow or orange dye, and on the right we have a lesser amount of purple dye. Well, over time, what's going to happen is the yellow is going to diffuse toward the right until it reaches equilibrium. The purple is going to diffuse to the left until it reaches equilibrium. At the end, we would have a mixture of equal amounts of purple and yellow on both sides. And the purple is unaffected by the yellow and vice versa. So each one of these materials will diffuse down what we're going to call the concentration gradient, and they're unaffected by the concentration gradient of the other material. So what determines the direction of travel? Well, it's what we call the concentration gradient. And that basically is the area along which the density of the material increases or decreases. And the material is always going to move from a high concentration to a low concentration. So going back to our perfume bottle, when you uncap that perfume bottle, right there in that corner of the room, there is a high concentration of perfume molecules. The opposite corner of the room, there is a very low concentration. And the concentration gradient is this area where the density of molecules changes. And so as those molecules spread, they are moving what we call down the concentration gradient from high to low concentration. This doesn't require any work to be done to move down the concentration gradient. It doesn't require energy. So this is passive transport. So materials that move across a membrane down their concentration gradient from high to low concentration, that's called passive transport, because the cell doesn't have to put out any energy for that to happen. You've probably heard of this other word called osmosis in relation to diffusion, and osmosis is simply diffusion of water. Because water is so important to biology and cells, we have a separate name for diffusion of water, and it's called osmosis. So osmosis is the diffusion of water across a selectively permeable membrane. I don't know if any of you guys ever watched a cartoon. It was called Osmosis Jones, kind of a funny cartoon live-action movie about the human body. If you've never checked that out on Netflix, you might want to grab that. It's pretty funny. It's got Chris Rock is the main character. He's a white blood cell inside the body, and it's just kind of funny. It's called Osmosis Jones. Anyway, back to osmosis. Water diffuses across a membrane from a region of lower solute concentration to the region of higher solute concentration until they are both equal. So let's take a look at what that means. Your book, I can't remember the page number, but it's figure 7.14, has a great example of how you can see this in a laboratory. So let's say you have this nice U-shaped container, and it's separated by a selectively permeable membrane, and it's got water on both sides and sugar molecules on both sides. On the left side, you have a low concentration of sugar, so you only added, say, 1 teaspoon to the left side. To the right side, you added a high concentration, say it was 2 or 3 teaspoons of sugar. So you have a high concentration of the solute on the right, low concentration of solute on the left. Now let's say this membrane is selective. In other words, it doesn't allow everything to pass through. It will allow water to pass through, but it will not allow the sugar molecules to pass through. Perhaps the sugar molecules are too large. So what's going to happen over time? Well, if you give it enough time, you'll see osmosis occur. You'll see water begin to move across this membrane from the left to the right, and the water in the right tube will actually begin to rise. How does that happen, or what exactly is going on? Well, it's going to reach equilibrium. The concentration is going to reach equilibrium. So see how the concentration over here on the left is low and the concentration over here on the right is high? And then at the end, if you look carefully, the concentration is pretty much the same. There's the same amount of solute per volume of water. And that's because of osmosis. The free water molecules that are not attached and surrounding the little particles, they can pass through. But the water molecules that are kind of bound and attached to the solute cannot get through. So this selective membrane has openings so small that the water molecules themselves can pass through, but the sugar or the sugar particles with water surrounding them cannot get through. So the free water is able to pass. The occupied water that is attached to the solute cannot. So eventually over time you have more water pass through to the right, and you end up with an equilibrium where the solution has the same concentration on both sides. Let's take this to a more everyday example. I think I've mentioned before that I like sweet tea. Sometimes I go to restaurants here in East Texas. They don't have sweet tea. I don't understand. You're in East Texas. Everyone should have sweet tea. But at any rate, let's say we go to a restaurant that doesn't have sweet tea, and I put, say, 2 or 3 packages of sugar into my tea. A lot of that sugar falls straight to the bottom of the glass. And because the tea is cold, we don't get a lot of molecular action. We don't get a lot of motion and movement. Now, if I waited long enough, and it might take days, but if I waited long enough, those sugar molecules would dissolve in the water. At the bottom of the glass, the tea would be at a higher concentration of sugar. The top of the glass would be a low concentration of sugar. Over time, and again, because it's so cold and there's not a lot of motion, it might take a long time, but eventually there would be some osmosis occur. Now, we don't have a permeable membrane that we're putting in the tea glass, but even if we did, this would also show up. Eventually, you're going to have enough motion so that the concentration of sugar is the same throughout the entire glass. But that might take a long time by itself. Now, how can I speed up that process? Well, I can agitate the tea. I can stir it. That way I can get those molecules moving around quite a bit more, and I'll get that diffusion, and I'll get that concentration to even out quicker. Or another way to do that, of course, is to heat the tea, which basically just moves the molecules around more, and I can get that to dissolve a lot faster and reach a concentration equilibrium a lot faster. So it's the same effect. If we were to take that tea and I were to somehow be able to put a membrane halfway into the glass and all the sugar was at the bottom of the glass, well, what we would find is some of that water would move through that membrane to even out that concentration of sugar. If my amazing sweet tea example was not enough to understand, here's a quick little animation of osmosis. So this represents inside the cell, the light green. The blue represents outside the cell. The yellow line represents the cell membrane. And of course, these are little water molecules. And so right-click and hit play, and you can watch an animation about osmosis, which is the diffusion of water across a plasma membrane or a selective membrane. Now, as we know that water is so important to living creatures and so important to cells that, again, we made our own separate word for diffusion of water called osmosis, and this is a very important part of keeping cells alive. So let's look at cells that don't have cell walls first. So that would be like animal cells. The water balance of cells without cell walls. So we're going to look at a word called tonicity. Tonicity is the ability of the surrounding solution, like the material, the liquid outside of the cell, to cause a cell to gain or lose water. You would describe its tonicity in a couple of different ways. You could call it isotonic, you could call it hypertonic, or you could call it hypotonic. So those 3 words represent the tonicity of the liquid surrounding the cell. If it's an isotonic solution, That means basically the concentration is balanced inside and out. So the solute concentration is the same inside the cell as it is in the liquid outside the cell. This means that there's no net water movement across the cell membrane. That doesn't mean there's no movement at all. There is. There is water going out of the cell and water going into the cell, but it's about an equal amount going both ways. So if the solution that the cell is sitting in, the liquid outside the cell, is isotonic, if that describes its tonicity, well, you're not going to have a lot of water moving in and out of the cell, or better to say, you're going to have an equal amount of water moving in and out of the cell. So think of isotonic as being balanced. The next one we're going to look at is called hypertonic. Hypertonic means the material outside the cell is saturated with solute. It contains a lot of solute. So the solute concentration is greater than inside the cell. So the liquid outside the cell has a higher concentration of solute than inside the cell. What's this going to cause to happen? Well, that's going to cause water to move out of the cell to equal that concentration, to bring that concentration to an equilibrium. That's going to cause the cell to lose water. So if the cell is sitting in a hypertonic solution, in other words, the solution is heavy with solute, it is saturated with solute, that's going to cause water to leave the cell. The last one is called hypotonic. Hypotonic is the opposite of that. Hypotonic, you could think of a hypotonic solution as a dilute solution. It does not contain a lot of solute. So if the liquid outside the cell is hypotonic, that means the solution concentration is less than what's inside the cell. What's going to happen is water is going to move into the cell, again, to even out that concentration. And so you're going to have water entering the cell, or you're going to have more water entering the cell than leaving the cell. So isotonic is balanced, hypertonic is saturated with solute, and hypotonic is dilute. And all these three words represent the tonicity. That's the tonicity of the surrounding environment. What is the solution outside the cell? And then what does that cause the cell to do? Does it cause the cell to lose water, gain water, or is it at equilibrium? Of course, a picture is worth 1,000 words. So here's a great picture. This is a red blood cell, animal red blood cell, which doesn't have a cell wall. So if the animal red blood cell, let's just look at the top one right now because we're talking about the cell that doesn't have a cell wall. So just concentrate on the red blood cell at the top. If that blood cell is sitting in a hypotonic solution, in other words, the blue material around the cell is lower in concentration of solute. In other words, the concentration of solute is greater inside the cell. This is a dilute material outside the cell, that's hypotonic. Well, that's going to cause water to enter the cell. As a matter of fact, it may cause so much water to enter the cell that the cell bursts open like a balloon, and that's called lyse. So when the cell bursts, it's lysed. So if you have a red blood cell, and you can actually do this in the laboratory, and you put it in a hypotonic solution, so much water enters that cell that it may burst like a balloon. In the middle we have the isotonic solution. The isotonic is the balanced, so the cell has its normal shape. You have water leaving, you have water entering, exiting, but it's the same amount basically going in as out because the solution is at the same concentration inside and out of the cell. Finally, you have what's called a hypertonic solution. If you put the cell in a hypertonic solution, in other words, the outer liquid is heavier in solute, it's more concentrated in solute, you're going to have water leaving the cell. And you're going to have so much water leave the cell possibly that it shrivels up and deforms and in some cases will kill off the cell. So you can do this in a laboratory too, where you take a red blood cell, you stick it like, say, in a saltwater solution, which is hypertonic, and you'll have water leave the cell until it shrivels and dies. So this can be a problem for living creatures because their environments tend to change. The cell's environments tend to change. Sometimes it's in a hypertonic solution, sometimes it's a hypotonic solution. So they have to have the ability to regulate this. Otherwise, you would have cells shriveling and bursting all over the place all the time. So the word for this is osmoregulation. Osmoregulation is controlling the solute concentration within the cell. So you can help control the water balance in and out of the cell, and you have to do that to stay alive. And a great example is this little single-celled protist, a paramecium. You're probably familiar with those guys. They live in a hypertonic solution of pond water. So that means the solute concentration inside themselves is higher. So water is always rushing in. Their cell bodies are constantly taking on that pond water because the pond water is hypertonic. What that means is they've got to get rid of that extra water or they're just going to blow up like a balloon and pop. They actually have a vacuole that we talked about previously, a contractile vacuole. It acts like a pump. And so they will bloat up with water, and then that vacuole will collect the water, go to the cell membrane, fuse with the cell membrane, and purge that water and pump that water out. So in this picture, a little round area right there is actually the contractile vacuole. So basically, this little guy has to constantly bail water out of his body. Because he is constantly taking in water due to osmosis, he has to bail it out using a contractile vacuole. And there's a quick little video that shows that right here. So if you want to hit click, I don't know if you have to right click and hit play, or in the PowerPoint or Adobe file, if you'll just click on this, it'll play the little video. And you're looking just right in this area for a little circle that kind of opens and closes, and that's that vacuole purging or pumping water out of its body. So what about creatures that do have cell walls? Plants, obviously, their cells have cell walls, but the prokaryotes, the single-cell prokaryotes, the fungi, even some protists, they also have cell walls. So if you have a cell wall, it helps because a cell wall helps maintain water balance. And a plant cell actually kind of prefers to be in a hypotonic solution. It likes to be kind of swollen up with water. And so it will swell with water, taking in water from that hypotonic solution until it's what we call turgid, or very firm and rigid. And it doesn't burst, or certainly doesn't burst as easily, because it has that nice strong cell wall to kind of hold things in place. So imagine you have a water balloon inside a plastic square box. You can fill that water balloon and it will only stretch until it hits the plastic square box, and then it won't grow anymore. Unlike an animal cell, which is like a water balloon without the plastic box, you fill it with water and it'll just keep on taking on water until it bursts. So the plant cell prefers to swell a little bit larger and take in more water than the animal cell. As a matter of fact, if the plant cell is in an isotonic solution where there's no net movement of water, the plant cell actually becomes a little bit limp, what we call flaccid, and the plant starts to wilt. So like this picture down in the corner, if you didn't water your mom's plants for a few days, they may start to wilt. And that's because the plant cells are actually no longer turgid and rigid and firm. They're starting to get limp and soft. That doesn't mean that the plant is dead. It just means that the solution that the cells are in is isotonic, and that's not quite enough. The plant actually prefers to be taking in water and be in a hypotonic solution. So here's a nice little video of Elodea or Elodia. I've mentioned this plant before. It's an aquatic water plant, very good for looking at underneath the microscope. So this is what turgid Elodia cells look like. They are very filled with water. The cell membrane is pressed tightly up against the cell wall. And you'll also see some movement of the chloroplasts. Remember, that's called cytoplasmic streaming. We talked about that, I think, in Chapter 6. So plant cells actually prefer to be in a hypotonic solution where they are taking on water and swelling up to their max. In an isotonic solution, they actually begin to wilt a little bit. They may not die, but they will begin to wilt. How about in a hypertonic solution? Let's say that the solution outside the plant causes them to lose water. Well, in a hypertonic solution, you've got a problem because the cell membrane actually begins to shrink and it actually starts to pull away from the cell wall. And this usually is deadly. It's a lethal effect called plasmolysis. And so if you've got a plant and you haven't watered for weeks, that extreme wilting has probably resulted in cellular plasmolysis. And that poor plant is probably not coming back. He is probably dead and gone. And then if you water him now, it probably is not going to do any good. So down here on the right is a plant cell in a hypotonic solution, taking in water, nice and happy and fat. Here you have a plant cell in an isotonic solution. It's not quite as happy. There's water coming in and out at an equal rate, and it's what we call flaccid. Notice the cell membrane doesn't butt up against tightly to the cell wall. And then over here we have a hypertonic solution where the cell is losing water. Big time problem. The inner part of the cell membrane shrinks down, pulls away from the cell wall, and we call that plasmolysis. This cell has plasmolized and will probably not be coming back. It is dead and gone. Here's a great video taken from a microscope from the Elodea or Elodia cells. You can do this if you take a Elodia leaf and you place it in a saltwater solution, and that creates plasmolysis, and you'll see the cell membranes pull away from the cell wall. The cell loses water, and these cells are going to probably die and not be recoverable. So if you just click on this, you should be able to play it. One last thing to talk about before we take another break. So remember we talked about diffusion being a passive process. When water molecules or other materials diffuse down their concentration gradient from a higher concentration to a lower concentration, that's what we call passive, because it doesn't require energy input by the cells. It's passive transport. But there is a form of passive transport that actually uses some of those transport proteins we talked about, and this is called facilitated diffusion. So if the material is just diffusing through the plasma membrane all by itself, that is one sort of diffusion. But if it's actually using some of these proteins to diffuse, that's called facilitated diffusion. And it's still considered passive transport because the net movement is down the concentration gradient, but it's going to use the help of these proteins to get across the plasma membrane. So in facilitated diffusion, the transport proteins that we talked about, they help speed the process along. They speed the movement of these molecules across the membranes. So remember we talked about channel proteins. Channel proteins just provide a tunnel or a pathway for specific molecules or ions to cross the membrane. We talked about the aquaporins. They are a channel protein that allows for the diffusion of water. And there's another type of passive transport protein, a channel protein called an ion channel. And an ion channel opens or closes in response to a particular ion. So it also can allow these ions to pass through, but they're doing so down the concentration gradient, so it really doesn't require energy from the cell. So at the top here we have a channel protein. So notice that the concentration of this particular molecule, the brown ball represents the molecule, it's higher outside the cell than it is inside the cell. So it's going to be diffusing into the cell to even out that solution, or to even out that particular gradient, and it's going to use this protein as a pathway, a channel, to just make it easier to travel through to the inside of the cell. The other type of transport protein we talked about, remember, was carrier proteins. Carrier proteins bind to the molecule, and that creates a change in their shape, and that moves the molecule from inside the cell, or I'm sorry, from outside the cell to inside the cell, or it could be from inside the cell to outside the cell. So carrier proteins also allow for diffusion, and that's going to be passive transport because it's still going down the concentration gradient, but it's just another method for moving that material. There's actually some diseases. Your book talks about a kidney disease called cystinuria, I believe, cystinuria, that's caused by a malfunction in this type of transport system. So people who have this disease, they lack a particular carrier protein inside their kidneys, and that allows for the buildup of a particular amino acid that leads to kidney stones and other problems. So a carrier protein transports this material across the membrane, but still it's passive transport, it's still diffusion, it's still not requiring energy. So here down at the bottom of this picture, here's a carrier protein. So again, the brown ball represents a molecule or some material that is higher in concentration outside the cell. So it's going to be diffusing toward the inside of the cell. And in this case, it's using a carrier protein to do that. The carrier protein binds to the molecule, changes shape, releases the molecule on the other side. Again, passive transport because the molecule is moving down the concentration gradient from an area of low concentration or higher concentration to lower concentration. Sorry about that. I actually clicked on the picture there. All right, we're going to stop right there. We're going to take a break, and then we're going to move on to active transport. So far we've looked at only passive transport, which doesn't require the input of energy. It's moving down the concentration gradient from high concentration to low concentration. But what if we need to do the opposite? What if we need to move materials up the concentration gradient? Well, that requires energy, and that's called active transport. That's what we'll look at in just a minute.